I'm Tian Tian. I'm the social media editor of Parkinsonism and Related Disorders Journal. And we are truly fortunate today to have with us Dr. Philip Pawlowski, who recently published an article in the February 2026 PRD entitled Altered Subthalamic Alpha Beta Oscillations in Parkinson-Associated Early Onset Parkinson's Disease in relation to off dystonia. These are exciting times in the field of advances in adaptive DBS, deep brain stimulation, and growing interest in identifying unique neuronal activity biomarkers. Well, today we shall learn more from Dr. Pawlowski. Hi, Dr. Pawlowski.
Hi, I'm very honored to be here. I'm Pawlowski, and thank you for calling me doctor, even though I'm just pursuing my PhD right now.
Okay, so could you briefly describe how the study was performed?
Sure. So our lab actually routinely performs analysis of microelectrode data and LFBs in patients with Parkinson's and dystonia. So Parkinson's is my specialty here. So at first, we just were curious in analyzing patients with early onset Parkinsonism because there's still no consensus in this field whether or not they are different in terms of their basal ganglia activity and whether or not they require specific DBS approaches. But then we thought about one more thing to add here, and we decided to add genetic stratification, which is a novel approach for our lab. So we just took the easiest way here and performed MLPA for plain screening for the most popular nucleotide variations associated with Parkinsonism. And that's where we got really lucky because out of 40 patients that we studied, 6 had one and the same mutation, which was homozygous deletion in Parkin gene of the same exon even. And here, actually, there is a possible limitation because MLPA is prone to some kind of false positive results. I mean that even though we report these mutations as full homozygous deletions in this exon, we bear in mind that this might have been homozygous deletion, but not of the whole exon, but maybe just of the binding region for MLPA reaction. But still, this mutation took place and it was homozygous. So we just moved on to compare these patients with some group of comparison, control group of patients with early onset PD with no mutations found. And we tried to select the patients with the closest PD onset, age, and PD duration to those who had mutations. So we just performed our ordinary analysis. We classified the neurons that we recorded during DBS operation in the STM. We classified those neurons into patterns, tonic neurons, burst neurons, and pulse neurons, depending on their activity. And we also specialized on oscillatory activity of both single cells and local field potentials. Because we expected some differences there. Very little is actually known about whether Parkin patients are different, but we found one paper by Whale and others who found statistically insignificant but present differences, and we were curious to investigate them.
Wonderful. How did the presence of dystonia influence the observed differences in STN electrophysiological activity between the Parkin-related early onset Parkinson's disease and the idiopathic early onset Parkinson's disease?
So at first, just like I said, we just compared Parkin patients with control group, and we found very few differences. And at that point, I was like, I can't believe it, because there ought to be differences, because Parkin patients are very different in terms of clinic. They typically have milder symptoms, their symptoms progress slower, they have very early onset typically, and they have no cognitive decline and no Lewy bodies aggregated in them. So there ought to be some differences. And then I noticed that in our groups, actually, some patients had dystonia, both patients with the mutation and some of those who were in the control group. And even though patients with simultaneous Parkinsonism, I mean bradykinesia, rigidity, and tremor, and dystonia are not exactly well studied, we don't know much about their activity as well. I had some preliminary data on the globus pallidus, another basal ganglia structure, that showed that actually there are differences. And I also expected some dystonia-associated activity to exist in the STM2. I decided to put these patients with dystonia aside and compare not the two initial groups, but 4 of them, accounting for dystonia. And I discovered that, yes, indeed, dystonia proved to matter in terms of alpha activity. But what I didn't expect was that that was true only for Parkin mutated patients. In fact, we observed that patients with this mutation and without dystonia didn't tend to exhibit oscillatory activity at all. I mean, they had some, but it wasn't that widely spread. It was concentrated in the high beta and around 20 Hertz. And some of the single unit peaks were also found in theta, but not in alpha range at all. At the same time, Parkin patients with dystonia, they had many single cell oscillations in the alpha range. And in fact, they had even more oscillatory peaks and even more coherent pairs than all the 3 other groups, more than all control patients. So on the other hand, we have a situation where when Parkin patients without dystonia don't seem to exhibit oscillatory activity, at the same time, Parkin patients with dystonia oscillate. At the same time, control patients had their single cell oscillatory peaks really evenly distributed, regardless of the dystonia. So what can we see from this? On the one hand, it seems that at least in some patients, and for some reason these are Parkin patients, alpha activity seems to reflect dystonia. Actually, as expected, but still interesting. At the same time, in patients without this mutation, in control patients, alpha activity is also present, but it doesn't seem to reflect dystonia, at least in full, because some of them don't have dystonia. But what we also found that single cell patterns weren't equally distributed in these groups. And while I expected that patients with dystonia should have more pulse and burst cells, just because their overall firing rate should be lower, as predicted by firing rate and firing pattern models of Parkinsonism and dystonia. That was true, but I also found that Parkin patients have similar distribution. They also have a lot of pulse and burst cells, even without dystonia. And this may mean that these patients are in fact somehow different I mean, in how their basal ganglia and how their brain structures interact with each other, because their patterns seems to be different.
Very well designed study, and also very interesting findings. I was wondering what might explain the reduced alpha and low beta oscillations, but the increased pulse burst neuronal patterns in the Parkin patients without dystonia.
Oh, this is a tremendously difficult question to answer, and I don't know the answer. So let's speculate a bit. So at first, let's discuss the patterns. As I said, it seems to me that patients with mutated Parkin have their brain regions either communicate in some different manner compared to control patients, or have their neurons, I mean separate single neurons, they have them like changing their biophysical properties. I mean, it seems plausible that their neurons generate spikes differently, perhaps having different threshold potentials or something like that. And actually, both these ideas have some grounds, but both are really understudied. At first, there are some studies, and I mentioned them in the paper. The first one describes that in human neurons that were artificially generated from human pluripotent stem cells, and these stems were taken from patients with double mutations in PRKN and PINK1 genes. These neurons indeed have unique biophysical properties. They answer interestingly and to excitatory potentials. They are overexcited easily. On the other hand, we know that mutations in PRKN gene lead to some alterations in cell metabolism, actually, in mitochondrial metabolism, and these can plausibly lead to some specific cell death scenarios. And this is also hypothesized in a number of papers, and it seems plausible that in patients with PRKN mutation, the scenario of neurodegeneration is somehow unique from other Parkinson disease patients. And this is how it is reflected on the single cell level. About oscillations, it's also hard to say because alpha activity is related to a number of phenomena, and among them are dyskinesias, tremor, let's say movements, including involuntary movements, uncontrollable movements. And our data partly provide evidence to this theory because in Parkinson patients, as I said, alpha activity seems to be related to dystonia specifically. On the other hand, we see that in control patients, it doesn't seem to be related to dystonia specifically, and it kind of doesn't make sense. I think that here we see that even one phenomenon can be actually different in its nature. We see it as alpha activity, I mean, as oscillatory activity in some range. But what we can actually see are perhaps different processes of brain regions communication. And about low beta, I also don't know why Parkinson patients doesn't seem to have low beta oscillations, even though they are, in fact, Parkinsonian patients and they ought to have it. The only idea and the only explanation that I have in mind is that we can remember that actually levodopa and some dopaminergic medication was shown and found to shift the oscillatory mode from low beta range to high beta range on animals. And we can suppose that in patients who struggle with the disease, with Parkinson's disease for a long time, these oscillations may shift naturally on the other direction, from high beta to low beta. It seems kind of logical if we remember that high beta is supposed to be prokinetic and low beta should be associated with bradykinesia and rigidity. Here, in our case, Parkinson mutated patients are known to progress slower, and this can make sense why they don't have these low beta oscillations. On the other hand, then, in our case, patients without dystonia from both groups have similar overall epidural score. So we cannot say that these absence of oscillations should be directly related to differences in bradykinesia and rigidity. But we can ask ourselves, is it possible that, in fact, low beta activity reflects not like these symptoms mechanistically? You know, maybe it reflects some broader pathological processes in the brain. This is to be studied, I guess.
Fantastic. Now, a practical question. How could the electrophysiological differences identified in Parkinson-related early onset Parkinson's disease inform a personalized DBS programming strategies?
Unfortunately, at this very moment, I don't see any way that they could, because we still know very little, too little, in fact. What I think is to be done here. At first, we should specifically analyze the VTA, the volume of activated tissue, in the target structure in patients with identified mutations. If we, for example, see that there are in fact differences in the clinical effect of DBS, depending on where the VTA is located, in the STM or maybe in the GB, then this result is handy for the clinicians just the way it is. You can adjust stimulation or the electrode location and get the desired effect. As a basic scientist, and I am one, I am more interested in comparing this data to patients without mutations. And I wish to see whether or not there will be differences. What I mean, for example, if we take some big cohorts of patients with mutations and without mutations, and have a look at their VTA location at the single cell activity in this VTA and at the clinical effect. For example, if we see that regardless of the mutation and regardless of the single cell activity, the clinical effect is the same for one the same VTA location, then we should say that perhaps it doesn't really matter whether or not this patient has a mutation or something else. We just should locate the electrode at this place, and we will be doing the best we can here. And also, that from my point of view, it may mean that the STM or the single cell activity in this region of the STM maybe isn't the main source of pathological information we should look for. On the other hand, if we found any differences, for example, if we see that this clinical effect, perhaps it will be different for different symptoms, like for tremor, for actual symptoms, for bradykinesia. If this effect depends on the VTA location and or on the single cell activity in this VTA, and these dependencies are different in patients with and without mutations, then this is to be studied further, because this is the way to look for personalized treatment, in fact. But overall, I should say that I don't currently believe that Parkin patients would benefit greatly from such studies, because as far as I know, patients with mutated Parkin are actually, they respond well to DBS treatment, as it is now. But there are some other mutations that are known to, empirically known to differ from this point of view from ordinary patients, from idiopathic patients. And I'm saying about GBA mutations, which cause great cognitive decline and some cognitive problems, DBS-induced cognitive problems as well. I'm talking about LRRK2 mutations here, because I've seen a recent paper pointing out that these patients, for some reason, need to adjust their stimulation settings after the operation, shortly after the operation. And generally, I believe that maybe these patients should be studied further from this perspective, because they are likely to benefit more from such kind of personalized treatment.
Awesome, I totally agree with you. Lastly, what are the limitations of the study and future directions?
Well, the first obvious limitation is the genetic method we used for screening. MLPA is, as I have already said, is prone to false positive results, and this is a big issue here. We are going to confirm our findings using the full genome sequencing sometime soon, and we are going to go on with this research using full genome sequencing only, because it's more reliable. And we will also be able to find some point mutations, and not only copy number variations, I mean. because MLPA is usually only able to detect differences in copy of number variations. We don't know this for all the patients in our study, but in patients with the mutation and in control patients, dystonia may be different in origin, because in some patients it was observed from the PD onset, and in some patients it only was observed some years later. And about future directions, we are going to analyze activity in the BTA, as I've described, and go on with a full genome sequencing actually.
A very beautiful paper. Thank you for joining us again today. And for those listeners, please read the full article in the Spotlight article, February 2026 Parkinsonism-Related Disorders Journal. And again, the title Altered Subthalamic Alpha-Beta Oscillations in PRKN-associated early onset Parkinson's disease in relation to off dystonia. Thank you.
Thank you.